Mr. Sonnenberg here. Today we're going to be talking about extending human vision. So this is for Science 8 students studying light and optical systems, topic 5 in your textbook. So there are two specific outcomes that we want to touch base on through this screencast, and one is I want you to understand how to explain how microscopes work and also understand how binoculars, eyeglasses, and telescopes function, and then also that you can explain new technologies that can enhance human vision. So we have done some work in a prior unit on understanding how microscopes work and understanding and labeling the various components. So we won't go too much. I'll breeze through that because we do have working knowledge on that. But moving on, one thing basically, if we were to take a magnifying glass and if I was just to draw it right here, so we have our handle and then we have our magnifying glass here. Okay, and then it's got its frame, and then there's a lens on the inside. Basically what a magnifying glass is, if I look at something, it's going to increase. So if you take a look here at this eyeball, the eyeball is increasing in size, and then also when we look at objects, we increase. So a magnifying glass is a very simple, basic microscope, and it magnifies up to about 10 times actual size, right? So if I was to put it in front of my thumb, magnifying glass, my thumb would look 10 sizes or 10 times bigger approximately. So in 1676 there was a Dutch scientist named Anton van Leeuwenhoek, and I have a lot of trouble saying that. I hope I said it right. Sorry. Maybe you guys should try that at home. It's not easy. But basically what he used was just a simple convex lens, and remember, convex lenses are going to bulge out. So we have a double convex lens that would look like such, right? That's convex. Right, so it's bulging outwards. It's thinner at the tops and thicker in the middle. And what he did was he used that simple convex lens to actually view bacteria, and he magnified it about 280 times, so he could actually see those bacteria. So basically, what microscopes look, or how they work, if we just take a look here, they actually have an eyepiece, okay, and or an objective lens, and this lens is going to actually help us see. So the objective lens would be right here, and that would be these three components that we're looking at in this area. The eyepiece up top has a lens as well that we look through here, but the objective lens actually forms the real image of the object, and then it's actually going to magnify. It's going to be magnified by the eyepiece as well. So we're forming the image through the objective lens, allowing light to enter through the tube. And what happens is then we actually can view the image up in our head. So usually what we have is, if you see here, we have a revolving nose piece on these compound light microscopes. And this revolving nose piece right here, actually you can twist. So I can turn that around, okay, and around. And what it does is it moves these little lenses here, here, and here, and we can actually magnify more or less based on those objective lenses capabilities. So we have three objective lenses that are going to allow specific amounts of light through those lenses that is going to capture the light to form that real image. Okay, so we can improve sharpness. We also have, if you take a look down here, if you remember this from earlier, we have coarse adjustment and we have fine adjustment. And the coarse and fine adjustment is going to adjust the height of the stage and whether it's close to the lens or farther away, and then that's going to help us to magnify. Also, it's going to help clarity. It's going to help us to clear the image up so we can recognize what it looks like. Down at the base here we have a diaphragm, and the diaphragm is going to control the amount of light that is actually portrayed through the stage, and so that we can see the image as well. And then we have a light source at the base, which is going to be right here. This light source is going to shine light upwards through the diaphragm, up through the stage, and then we can view the image. Up top through these objective lenses, up through the tube, up to the eyepiece, into our eyeball, which is all beautiful with eyelashes and a pupil, and then eventually back to our brain. All right, okay, that's a brain. It's green. Well, brain's not green, but and then it forms that mental image in our head. So that's how microscopes work. But we get a lot of our scientific knowledge from these extended human vision, or these concepts of extended human vision. So we could actually look at cells, and we can see what cellular activity looks like and such. And I'll talk about next how that's actually going to be of assistance to us. So we've learned a lot of things from the development of microscopes and telescopes, and earlier in the screencast I talked about telescopes and how we could see the galaxy and we could see images on the moon and such. But when we talk about microscopes, we can actually look at living tissue, and we can see that that living tissue is composed of cells. Now remember, we talked about that. We talked about cells building tissues and layers of tissues building organs, and organs working in conjunction with other components building organ systems within our body, right? And that's how that's our structure, and then that's how we can function. So these living tissues composed of living cells, they function and reproduce, and reproduction can be viewed, and we can actually view how these cells multiply along the way. And we could look at, say, cancerous growths, and we can use the microscopes to discuss how we can destroy viruses and how these cells they react and to both positive and negative stimuli as well. And we can also understand our genetic makeup as humans, right? And also that of plant plant vegetation and organisms, and then also other animals. But then we look at telescopes and some of the new discoveries in telescopes. Well, they keep getting bigger and bigger, and the reason they get bigger is we can attract more light. We can take in more light through the objective lens. Therefore, we can create images that are farther away and in real images, because the lens size, right, the objective lens which takes in the light, and we can form these images from greater distances. And that just basically opens up this huge galaxy and universe for us just to look at in the sky, and we can try to look farther away to pick up and learn more about what's out there, which is, you think about it, it's quite fascinating. But you have to remember one thing, though: the telescopes and microscopes, they do have their limitations, and that is revealed by the nature of light. We see shadows, right? A telescope can't see through or around corners. Nature of light states that light travels in straight lines, and it's absorbed by surfaces. Same thing applies. So even though we're looking through a telescope, we're still going to see that the nature of light is revealed, and it will have its limitations, definitely, because we are going to see shadows, and the light is going to be absorbed, and we're not going to capture all the light. So again, with every discovery comes limitations, but we keep advancing, and technology and science keeps advancing, and we see some of these we see things, and we know information about cells, and we've seen planets, and we get all these images because of this technology and how it keeps advancing, and there's new discoveries. So anyways, guys, that's extending human vision. We've talked about microscopes. We talk about some of the components and new discoveries that we've seen and how they help us in society. And also we see how microscopes can really help us to take a look and dissect ourselves through vision and see what our cells look like, how they act, how things are formed within our body. So pretty interesting stuff. Hopefully this has helped. And hopefully you've taken some notes and jotted down some notes. Remember, if there's some parts of this that you didn't understand or didn't catch, you can play, pause, rewind, and you can watch it again. Okay, thanks guys. Bye.